Hello, my name's Olivia.
I'm Jada.
My name's Katie.
And I'm Chloe.
So as humans nowadays, we live pretty overstimulating lives, right? We have phones and multiple screens constantly in our faces. We multitask like it's our job, and we do all of these things like it's just a part of life. Well, finding out what overstimulation does to the brain is very important to be able to relate it back to the way humans are living and its effects it may have on our psyche. So that being said, we're going to be discussing today the influence of habitat overstimulation on memory and hippocampal BDNF levels in mice. So a little bit of background. Brain-derived neurotrophic factor, or BDNF, is a protein that plays an important role in neuronal growth and survival. This protein is seen in effect with depression and anxiety, as well as memory and learning, which is what we focused on throughout our experiment. BDNF found in the hippocampus enhances long-term potentiation, or LTP. LTP is an elemental mechanism for memory and learning, and the best way it's measured is to examine its synaptic plasticity. This is related to BDNF in the way that the availability of BDNF greatly affects the plasticity of synapses, as well as the process of synaptogenesis. Environmental enrichment is a treatment where the hippocampus is stimulated through the placement of toys in the mice's cages, which is invariably their environment. With this process, higher levels of BDNF are typically found and are consistent with enhanced LTP, neurogenesis, and synaptogenesis. However, too much of a good thing can become a bad thing, and in this case, overstimulation is a result of enrichment being exhausted to the point of stress. In the brain, stressful events lead to activation of a neuroendocrine pathway known as the hypothalamic pituitary adrenal axis, or the HPA axis. Through the promotion of this pathway, cortisol is released, which then has opposite effects on BDNF levels in the brain. This decrease in BDNF leads to decreased LTP, which results in decreased memory. Taking all of this into account, we decided to look at the effects of overstimulation has on the mice BDNF levels and memory capabilities in our own experiment. Now Katie's going to talk about methods.
Thanks, Olivia. So diving into our methodology, so we used 10 CD1 mice that were split into both experimental and control groups. So each group had 5 mice. And then right after receiving the mice, we gave them an acclimation period of approximately 22 days just to adjust to their environment and where they were going to be living for an extended period of time. And then starting testing, so the experimental mice were subjected to what is known as habitat disruption, which Olivia talked about a little bit. Really, over the course of 7 days, we really just changed the mice environment to have different toys and different things covering the ground. And so they had to readjust each and every day, which we were hoping to induce overstimulation with. And then the control mice had a standard type 3 cage setup. And so they just, they had one house in their cage and that was it. We didn't want to affect the results by also overstimulating them. And then we chose the Y maze for our behavioral paradigm. So each mouse was allowed to explore the Y maze for approximately 7 minutes, and there's a picture of the apparatus in the next slide. And then after testing, mice were immediately taken to the physiology lab for cervical dislocation and excision of the hippocampus. So here's a timeline of events like I just explained, excuse me. So days 1 through 21 was our acclimation period, and then 22 through 32, like I said, control was just the type 3 cage, and then we had our overstimulation for our experimental mice, and then Y maze and cervical dislocation. So a little more about the Y maze. We used the Y maze as a test of working memory, and so like I was talking about, methodology followed standard procedure. Each mouse was placed at the end of arm 1 and was allowed to explore for 7 minutes. And then we expected mice to complete a series of alternations. Mice are inherently curious animals, and so we expected them to explore the novel areas as opposed to running to one arm and then going back to another, because that would be an indication of poor working memory capacity. So quantifying working memory, we observed the number of alternations, the number of errors, and the latency to complete the first alternation. So we define an alternation as choosing the novel arm as and moving forward in either a clockwise or a counterclockwise rotation. So there are 3 arms. We expected them to go 1, 2, 3, or 3, 2, 1, or whatever, as long as they went to 3 arms consecutively. And then an error was defined as re-entering the same arm as opposed to choosing the novel arm. And so again, going maybe from arm 1 to arm 2 and then going back to arm 1, that would be considered an error in our experiment. Behaviors were tracked following testing using the Behavioral Observation Research Innovation software known as BORIS. And then an overview of the analysis of our results. So we used an ELISA, which is an enzyme-linked immunosorbent assay, and then also a standard Bradford assay. And both of those together they allowed us to calculate the BDNF to protein ratio. And then we also used an independent 2-tailed t-test, assuming unequal variances, to measure the latency to complete the first alternation and the number of errors. And with this, we were able to calculate a p-value that would tell us whether or not the results were statistically significant. And in these, we compared the experimental and the control groups. And then we also ran a repeated measures ANOVA, which was an analysis of body mass over time, just to make sure that the change in weight over time between groups and between weeks didn't have any effect on our results.
So here in figure 2, we can see that the bar graph is showing the latency of both groups to complete the first alternation of the Y maze, like Katie mentioned earlier. So the error bars are just showing standard error of the data. And we found that the control group had a higher latency than the experimental group, which was the opposite of what we hypothesized. And we're going to be discussing this a little bit later in our discussion. But this basically just means that habitat overstimulation did not significantly affect memory as we thought it would. So next in figure 3, we can see that this bar graph is showing the average number of errors for each group. Again, the error bars are showing standard error of the data. And also, again, we found that the control group had more errors than the experimental group, which was the opposite of what we hypothesized. And again, we're going to be discussing this later.
Since we were subjecting our mice to habitational stress, or at least for the experimental group, we took the body masses that we calculated over the 5 weeks we did testing, and we compared them between the weeks and between the groups by doing a repeated measures ANOVA. And this ANOVA returned a non-significant p-value between the groups, which means that the weights between the weeks for the experimental and control mice did not differ, which is kind of surprising, and I'll talk about that in a minute. And then between the weeks, they were significant, meaning that there was a visual fluctuation between both groups, between weeks 1 or 2, or between weeks 2 and 3. This is sort of surprising because if we are subjecting the experimental mice to stress, we would expect possibly that their weights might be, depending on the type of stress, maybe lower or higher than the control group. So the fact that they are the same based on the p-value that was returned is a little concerning for us. So that means that the overstimulation may not have had a significant effect, or possibly there's something going on also with the control group. And we'll talk about that more in the discussion.
So in figure 5, we have the average number of picograms of BDNF per microgram of protein in mice hippocampi. And we found this using the ELISA assay that Katie mentioned earlier. So the error bars are showing standard of the data, error of the data again. And as you can see, the bars are about the same level. And so that just means that habitat overstimulation did not significantly affect BDNF levels as we thought that they would.
So because we saw a difference between the latency and number of errors and the p-values returned for the BDNF to protein ratios, we decided to do a correlation regression graph comparing latency in figure 6 and the number of errors per mice in figure 7 to these ratios. As you can see for both graphs, they look very similar. Each point on each graph was a representation of the individual mice, individual mouse used. And you'll notice for figure 7, we only had 4 mice for the control group due to a technical error that we had, but we were able to get all 5 for the latency. And for both groups, even though there was a noticeable difference between the latency and number of errors, the level of BDNF to protein ratio stayed relatively the same for both. The correlation factor for the latency to BDNF was 0.142, and the same value for the number of errors to BDNF ratio was 0.149, which is incredibly weak. We would need to see an R value of probably 0.5 or higher, closer to like 0.75 or above, to actually determine a correlation between these two. All right, so based on the results, we're going to go over a little discussion with you guys. So we expected that the experimental BDNF to protein ratios would be lower than the control due to being exposed to stress, and we would eventually see that in our behavioral tests. However, we observed that the experimental ratios to the control were practically the same. In fact, returned a p-value of 0.95, which is very, very close to 1, which means there would be absolutely no difference between the two. And then with the behavioral test, the Y-maze, we expected that the experimental latency times and errors would be more than the control. However, we observed the exact opposite, as Jada mentioned. And we actually saw that the experimental latency times and errors were lower than the control. And we have an asterisk here because the number of errors actually returned a p-value of 0.068, which would be a strong trend. But since we had to occlude one of the control mice, we think that if that were included, it possibly could have lowered that p-value to a significant value. So we decided to include it as a significant value here. So why would we see not a significant difference between, say, the BDNF to protein ratios? We came up with a couple of scenarios that might be affecting these. For one, the experimental mice may not have actually been overstimulated. It might have actually been similar to the environmental enrichment that Olivia talked about in the beginning of our presentation. And this could have increased the BDNF to protein ratios seen in the experimental mice that were then lowered back down by external stress because our experimental mice actually had a bully in it. So if the bully wasn't there, then possibly we could have seen that the experimental BDNF to protein ratios were actually significantly higher than the control. And we decided that this might have been a possibility because there's actually no standard for overstimulation. There actually needs to be more research to figure out what point of giving these mice toys and stuff to interact with, do they actually become overstimulated as opposed to maybe say environmentally enriched. The second scenario we had was that the control mice actually were understimulated. Although they didn't have a bully, we only provided them with one house. So they only had the bedding and their food and water to interact with besides this house. And they actually stayed huddled in it most of the time. So since they might have been understimulated, it's possible that we lowered the BDNF to what the expected experimental levels would have been. So this might have been why we saw that the two levels were relatively the same between the groups. And it's more possible that there was a combination of these two scenarios that led to that. A third scenario that's slightly different, we only tested the hippocampus BDNF levels for this experiment, but it is possible that the prefrontal cortex, PFC, might have had something to do with the differences that we saw. Spatial working memory, which we tested with the Y-maze, is actually processed using both the hippocampus and the prefrontal cortex. But for the sake of our experiment, we could only test the hippocampus. So the levels may have actually differed between the areas of the brain and the groups. And this would account for the lack of correlation that we saw between hippocampal BDNF and the groups. But since we didn't test the prefrontal cortex, we don't know. So here are a couple caveats that we had to our research. I already mentioned the prefrontal cortex BDNF concentrations not being measured and the no standard being available. But another one I wanted to mention was that we had to use the spontaneous alternation method. We tried using a different method for doing the Y-maze, which is called rewarded alternation. And this is where we would train the mice to go after a reward and therefore choose a particular arm or another. It still uses the same principle of novelty, but it better demonstrates hippocampal and prefrontal cortex spatial memory differences than say the spontaneous alternation would. However, we were unable to get our mice adjusted to the reward we had available, and for the sake of time, we stuck to spontaneous alternation. All right, this concludes our presentation. We wanted to give a special thank you to Dr. Waters, our professor in the Department of Biological Sciences, for the opportunity to conduct our research within our class. And then we have just a brief list of our references.